Bismillahirrahmanirrahim. Assalamu alaikum. Our today's topic is about bone. Bone is a highly vascular connective tissue filled with calcium salts and continuously being remodeled. Functions: bones give shape and support to the body and resist all forms of stress. Number 2, they provide surface for the attachment of muscles, tendons, ligaments, etc. Number 3, the skull, vertebral column, and thoracic cage protect brain, spinal cord, and thoracic viscera. Bone marrow manufactures blood cells, white blood cells, red blood cells, and platelets. Bone stores 97% of the body calcium and phosphorus. Now, types of bones. Bones are classified in terms of their shape and have been divided into 4 categories: long, flat, irregular, and short bones. Long bones. It has an elongated shaft, diaphysis, and 2 expanded ends, epiphysis. The shaft typically has 3 surfaces separated by 3 borders: a central medullary cavity and a nutrient foramen directed away from the growing end. Examples are typical long bones like humerus, radius, ulna, femur, tibia, and fibula. Miniature long bones, which have only one epiphysis like metacarpals, metatarsals, and phalanges. Modified long bones, which have no medullary cavity like clavicle and body of a vertebra. Short bones, their shape is usually cuboid, cuneiform, trapezoid, and scaphoid. Examples are carpal and tarsal bones. Types of bones. Number 1, flat bones. They resemble shallow plates and form boundaries of certain body cavities. Examples are bones in the vault of the skull, ribs, sternum, and scapula. Number 2, irregular bones. They do not resemble long, short, or flat bones. Examples are vertebra, hip bone, and bones in the base of the skull. Number 3, pneumatic bones. Certain irregular bones contain large air spaces lined by epithelium. They make the skull light in weight, help in resonance of voice, and act as air conditioning chambers for the inspired air. Examples are maxilla, sphenoid, ethmoid, frontal bones. Sesamoid bones. They are bony nodules found embedded in the tendons or joint capsules. Examples are patella, pisiform, fibula, etc. Functions of the sesamoid bones are to resist pressure and to minimize friction. These are the structure of bones. This is skull, mandible, clavicle, scapula, humerus, ulna, radius, carpus, metacarpals, and phalanges. Femur, patella, tibia, fibula, hip bone, etc. Types of bone tissue. Compact bone. Compact bone forms the outer surface of bones and consists of Harvansian system. Harvansian systems are formed by tiny concentric plates of bones called lamella, which surrounded Harvansian canals like the rings of a tree trunk. Each canal contains blood vessels, nerves, and lymphatic vessels. In compact bone, the lamella and Harvardian systems are packed closely together with only small spaces between the lamella to house osteocyte and between each Harvardian system for limbs. The microscopic structure of compact bone. Spongy bone. Spongy bone fills the substance of bone and also consists of Harvardian systems. Spongy bone differs from compact bone in that the Harvardian canals are larger and there are large gaps between the lamella. The spaces this creates are filled with red and yellow bone marrow, which is a mixture of fat and red blood cells. The spaces also helps to reduce the weight of the bone. The microscopic structure of spongy bone. And B. Electron micrograph of spongy bone. Gross structure of an adult long bone. Naked eye examination of the longitudinal and transverse sections of a long bone shows the following features. Number 1. Shaft. From without inwards, it is composed of periosteum, cortex, and medullary cavity. Periosteum. It is a thick fibrous membrane covering the surface of the bone. It is made up of an outer fibrous layer and an inner cellular layer which is osteogenic in nature. Cortex. It is made up of a compact bone which gives it the desired strength to withstand all possible mechanical strains. Medullary cavity. It is filled with red or yellow bone marrow. At birth, the marrow is red everywhere with widespread active hemostasis and is replaced by yellow fatty marrow with no power of hemostasis. Red marrow persists in the cancellous ends of long bones. In the sternum, ribs, vertebra, and skull bones, the red marrow is found throughout life. The two ends. In the long bone, they are made up of cancellous bone covered with hyaline cartilage. A mature long bone. Partial section of mature long bone. Now part of a young bone. A typical long bone ossifies in three parts. The two ends form secondary centers and the intervening shaft from a primary center. Before ossification is complete, the following parts of the bone can be defined. Epiphysis, the ends and tips of a bone which ossify from secondary cells are called epiphysis. Diaphysis, it is elongated shaft of a long bone which ossifies from a primary center. Metaphysis, the epiphysis ends of a diaphysis are called metaphysis. Metaphysis is the zone of active growth. Epiphyseal plate of cartilage. It separates epiphysis from metaphysis. Proliferation of cells in this cartilaginous plate is responsible for lengthwise growth of a long lawn. After the epiphyseal fusion, the bone can no longer grow in length. Now, blood supply of long bones. Nutrient artery. It enters the shaft through the nutrient foramen, runs obliquely through the cortex and divides into ascending and descending branches in the medullary. The nutrient artery supplies medullary cavity inner two-thirds of cortex and metaphysis. Periosteal arteries. They are especially numerous beneath the muscular and ligamentous attachments. They ramify beneath the periosteum and supply the outer one-third of the cortex. Epiphyseal arteries. They are derived from periarticular vascular arcades found on the non-articular bony surface. Metaphyseal arteries. They are divided from the neighboring system vessels. They pass directly into the metaphysis and reinforce the metaphysis branches from the primary nutrient artery. Now, development and ossification of bones. Bones are first laid down as mesodermal connective tissue condensations. Conversion of mesodermal models into bones is called intramembranous or mesenchymal ossification, and the bones are called membrane bones. However, mesodermal stage may pass through cartilaginous stage by chondrification during second month of intrauterine life. Conversion of cartilaginous model into bone is called intracartilaginous or endochondral ossification, and such bones are called cartilaginous bones. Ossification takes place by centers of ossification, each one of which is a point where a laying down of lamella bone formation starts by osteoblast. The centers of ossifications may be: the primary centers appear before birth, usually during 8 weeks of intrauterine life. The secondary centers appear after birth. Many secondary centers appear during puberty. A primary center forms diaphysis and the secondary center forms epiphysis. Fusion of epiphysis with the diaphysis starts at puberty and is complete by the age of 25 years, after which no more bone growth can take place. The law of ossification states that secondary centers of ossification which appear first are last to unite. The end of a long bone where epiphyseal fusion is delayed is called the growing end of the bone. Bone cells. Osteoblast, the cell responsible for bone formation. Osteocytes, these are the mature cells formed when osteoblast entrapped into a new bone. Osteoclast, the cell responsible for bone resorption, break down the bone. A fine balance of osteoblast and osteoclast activity maintains normal bone structure and function. Thank you very much. Allah Hafiz.